All right, thank you. And I will share right here. So thanks everyone for joining for this Ag Bio Data webinar. So today I'll be talking about how to stay grounded, or rather how to assemble structured biological knowledge with help from these large language models, or LLMs. So I want to start from possibly the broadest possible question any of this kind of talk can start with, and it's where does biological knowledge actually come from? We'd like to think that it's, at least in science, the result of repeated observations. Um, but at the same time, it's also learning from those observations, which we recognize that is in a task in itself. For biocurators, you know exactly how challenging this is, but even as a researcher, reaping the benefits of biocuration, you know that learning from the observations that are published in the literature is its own very laborious task. But it can, of course, be automated. Now, of course, it's really easy to say that and imagine all the potential ways that we can automate learning from any collection of text. But how would we actually automate learning from literature, knowing that there's a lot of literature out there containing a lot of different types of findings? Once we find those findings, how do we actually compare them in an automated way? How do we say, okay, well, this one is a more confident finding than another, or this finding really means the same thing as what this other paper said? Uh, and maybe at the same time, how do we integrate those different kind of observations that we find when we know that we're working with different studies or even replicates of the same studies? How do we integrate across different knowledge bases or across literature and different knowledge bases? How do we integrate observations across completely different fields or disciplines of those fields? If we find something really interesting in one particular model organism, uh, how do we see how that same type of phenomenon may or may not exist across the tree of life? If we observe that something happens, say in switchgrass, and we know it's a really highly conserved mechanism, uh, how do we explore, even compare that to the same kind of things that might happen in say the mouse or in E. coli, uh, or even in instances where people may have engineered those particular model organisms to have similar phenomena? And if we're talking about similar concepts, how do we even combine observations across them when they're defined in different contexts? We know that people can talk about the same types of proteins or the uh, same types of biological phenomena in completely different contexts. Sometimes it's for industrial applications, sometimes it's for agricultural applications, sometimes it's just basic science. So how do we integrate the observations that we have across those different levels? Uh, well, broad questions, and in this case, I'm going to give them a very simple answer, which is we need structured data. That's half the battle, at least. We need consistent data models, standards, and ontologies, which certainly help, but they don't actually do the entire work of structuring all the data that's available out there for us. So let's narrow this problem down a little bit more and say, how do we curate structured data from unstructured text? Traditionally, this has been the domain of human experts. It's a famously limited resource, although very effective in the long run, and it's one of the resources that we probably trust more than anything else. But over time, various people working in the field of biological data extraction have produced things like these rule-based extractors, like the SemMedDB project. Uh, these are largely projects where you just say, okay, well, I have a set of predefined rules, and given a piece of text, I want to extract everything that matches those rules, which can afford some fairly high precision, but unfortunately low recall because it's subject to how flexible those rules are. Uh, we can also find that the efficacy of these approaches can vary by domain and the exact structure that you're trying to extract. Some rules just afford themselves to certain concepts very well. If you're looking, for example, gene names, well, a lot of them are going to fit the same kinds of patterns that you may not see in something more complex like the exact name of a particular genome of a given species. So as time has gone on, folks have developed things like enrichment-based approaches. These are instances where you're defining the relationships between words and phrases a little more broadly and saying, okay, is there a particular term that shows up more frequently in a given body of text? Like if we're studying a particular disease, do we see the name of a particular protein show up more often than we'd expect? Or if we're looking at a particular model organism, is there a particular phenotype that has been studied a lot more broadly, which is one of the sources of potential bias in these approaches, which is that they are subject to publication bias. If something's just been studied a whole lot more than something else and you're looking for it in the literature, it can look like it's more important when in fact it's just more heavily studied. So a known often a bug more than a feature. So in more recent years, we've seen the rise of neural networks for natural language processing, and we've seen sequence-based approaches like the long and short-term memory models that rely on ingesting more or less fantastically large amounts of labeled text training data, essentially just treating it as a sequence and asking questions like, okay, well, given this sequence, what do we expect to be in front of it? Given, this 100 characters of text, what do we expect 101 to be? And unfortunately, the early advances in those recent years found that, okay, even if you give your model what seems like a really fantastically large amount of training data, well, it might still overfit for that particular training data. So then we saw kind of our first wave of language models, like if you're familiar with the BERT model. These transformer-based models, well, they still work with fairly large amounts of training data as their text. So in some ways, they're still kind of a brute force approach, but they give us one particular benefit, and it's that we can reuse these models quite well. They're very flexible, particularly if our problem is just avoiding learning the basics of language. With those LSTM models, much of that text data went towards just learning what language looked like. So if we have a model that more or less understands what the underlying logic of language, then we can reuse it and kind of further tune it for individual use cases. So I'd like to note the caveat here that each of these methods may still be very effective for some use cases. There's some use cases that where a rule-based approach is entirely appropriate and probably works very well. So the newer methods don't always perform the best. But for the particular task of translating unstructured scientific text directly into a structure, and here I'm going to phrase that as an arbitrary knowledge scheme or a set of schemas. What if there's a few things that we already know about those schemas that we're working with? Say they're very complex and they involve nested subclasses that we're trying to define. Say it's something like, okay, every relationship between concepts I and J, where I will be an object of type A and J will be an object of type B, but only from a particular set, and that's set C. I mean, these are the kind of things where we're defining data models that actually reflect the way that we want to be able to understand things in real world biology. And then furthermore, to make that data integratable again and integrate those observations, we want to be able to link to external unique identifiers like what we'd have in our existing knowledge bases. So can large language models like LLMs, and specifically things like GPT-3 and GPT-4, which have been very popular as of late, can they actually help? Well, one way we can look at this is by asking, okay, is bigger better? Because one thing that's nice about these large language models is that, well, they are larger than our previous generation, like BERT, and that's supposed to afford them the ability to have kind of emergent skills or these abilities to do things in kind of a zero-shot way, meaning you don't necessarily need to provide really detailed instructions or training data to have the model essentially understand what you're trying to get it to do. And benchmarks like this Big Bench language tech benchmark, which I believe was developed at Google, essentially ask that same question. In this case, they have 202 tasks and they ask, okay, well, do bigger models always do better? And well, results are mixed. Sometimes they do. For some tasks, they they actually do seem to have that emergent ability to do better, where you just hit a certain size in your language model, and the model is just able to accomplish those tasks. In some cases, the ability of your model to accomplish those tasks increases accordingly with the amount of additional parameters that it has, with the overall size of the model. In some cases, it actually gets worse. So what's really interesting are these emergent behaviors, this ability to just suddenly become much better and suddenly turn something that was previously seemingly impossible into something that the model can actually accomplish. So we'd like to think that this applies to most use cases, and well, in a perfect world, maybe it would. We'd have the solution to all of our data curation and integration problems. However, especially in the case of GPT, much of the training data that goes into these models is often very unclear, or may actually include fictitious claims. There's very little that goes into the training of these models that necessarily verifies that everything going into them is the real truth. That would be a very challenging curation problem in itself, but perhaps necessary, because another feature / bug of these models is that they do have human-like performance, especially when we're looking at biology and biomedicine. They output results when they're generating text that look an awful lot like what humans would generate when asked to do the same tasks, but they do not have the ability to do human reasoning or provide any additional details that would enable a naive user to distinguish between that generated text and what a human would actually say. They essentially they passed the Turing test and then some. This is especially challenging because, well, yeah, they're still perfectly capable of saying things that are outright false, like a recent study that was attempting to evaluate GPT-3's ability to do medical triage on simulated patients. And one thing that they noted in the course of the study was that GPT-3, when asked about its opinion on vaccines, responded directly with vaccines are not 100% effective. Vaccines can cause serious side effects. Vaccines can cause death. And it went on for quite a ways about, exactly how you should be worried about vaccines, which is not necessarily the opinion that you want your model to hold when you're also trying to ask it to triage patients. So this comes down to the idea of hallucinations, which has been kind of difficult to define for these large language models. But for many people, it just means an instance where the model is generating text that is essentially just grounded in language, but not necessarily in fact. It's perfectly capable of generating text that sounds true, or maybe even doesn't sound quite true, but at least sounds like language. But in many of those cases, it may not be grounded in any kind of fact at all, because that's not what it's modeling. Any modeling of fact is essentially a side effect of the process. So how do we address some of these limitations while still addressing that question of, how do we actually structure biological text? So in our group at the Berkeley lab, we've developed this method called SPIRES, or structured prompt interrogation and recursive extraction of semantics. You can also interpret that as information extraction that is grounded in reality. So the basic idea here is that we want to start with some kind of a knowledge schema, or you can think of that as your data model. This provides the basic structure that we'll use to assemble a query that we will pass to a large language model. Then, given some kind of input text, we feed that and the schema to our overall framework, which we call OntoGPT. That's all open source and online at the link shown below here. OntoGPT sets up a prompt that it sends to a choice of large language model. This may be GPT, or this may be another of a set of open large language models. Once it receives the completed text from that model, it searches a set of public databases and ontologies using the Ontology Access Kit, or OAK, which is also open source and accesses a variety of different knowledge sources. It uses that knowledge to ground the terms and relationships that it finds with unique identifiers present in those other resources, and essentially map them to a more consistent vocabulary. So what you get in the end is data that ideally matches the structure that you've set out in the schema, and has identifiers that can link through to those other resources. I should note also that this does it in kind of a recursive way, but I'll get to that a bit more in a second, because I want to mention something about kind of the overall use case that we envisioned back when we first developed this method. And it's that we thought this would be an ideal way to assemble knowledge graphs. One of the other things that our group is really interested in working with is developing these kind of reproducible processes for assembling and learning from knowledge graphs. And at its core, each of these graphs is really just a collection of relationships with this kind of subject, predicate, object. And that means if we have a consistent data model, for us, that's often the BioLink model, and a consistent set of processes for ingesting, transforming, and merging all that data into single graphs, then we can reproducibly develop knowledge graphs for a given field or given use case. So luckily, we also have a set of utilities and infrastructure that we call KGHub that's available at kghub.org to do exactly that. So in practice, what you can do is start with a given set of text and a well-defined schema. You can use that to output the structured data that you would use as just another input for a knowledge graph. And you can combine that with other established sources in graph form and what have you. So that's a work in progress, but we think that's the kind of thing that could be more broadly adopted, because we'd like to see more standardization across kind of knowledge graph technology as it is, especially in the biological and biomedical sciences. So at this point, sometimes folks will ask, why don't we just tell this large language model to extract all the relationships? Why do we need all this business with like grounding and data schemas? Like it's seemingly intelligent, or at least it does intelligent seeming things. Why don't we just tell it what we want? And well, in practice, you can kind of do that. So here on the left, I have some essentially randomly chosen text from a Texas page for young naturalists talking about predator-prey relationships. The text is a bit, I don't know, oddly threatening in places, but let's see if we can't extract something about the exact kinds of predator-prey relationships that we have in there. So I've essentially simulated that here by using OntoGPT and our SPIRES method to extract output from a language model, but I really just told it like, hey, extract all the kind of relationships that you find. I just want subjects, predicate, and objects. I don't really care what they are. And it does a reasonably good job. It extracts things like, okay, well, we found instances where this text talks about mountain lion stalking, killing, and eating deer. Okay, great. Found instances where mountain lion is using deer, human hunter is using deer. So, okay, we've got some semblance of a structure here. But we can improve that result by using a more defined relationship and by doing that grounding to a consistent set of concepts. So if we do exactly that, and instead we use a template that has been specifically defined to extract taxa, and here within the span of that schema, we've defined what we mean by taxa. We define the kind of relationships that we want to get out of the text, and we've even provided a few examples that the language model can use to provide extracted text that's more along the lines of what we're looking for. So we get some similar things, but it does narrow it down a little bit more. It says like, okay, well, human hunters have a target of deer, and here's the interaction type. In this case, they have several different interaction types of, well, they're stalking, shooting, and taking. So instances where this text is still largely extracted from our source text, which is what we expect, but it hasn't done a whole lot of that additional grounding that would really kind of make this data a little bit more integratable. We'd really like to at least say things like, okay, well, what are the NCBI taxon IDs of these individual species? So luckily we can do that. We can then pass it a schema that explicitly says, hey, let's try to align those taxa with NCBI taxon. Let's try to use Go terms for those processes and relationships. So what do we get then? Well, luckily in some cases, like when the text mentions deer, okay, well, we've got a corresponding taxon ID for cervidae. Okay, I'll take it. That's deer. It does miss things like humans because our language model chose to parse that as human hunter. And human hunter, unfortunately, does not show up in NCBI taxon. So this is a case where we might need a little bit more domain knowledge injected to figure out, we're really just talking about humans here. Also, instances like beef and pork, well, we don't have the domain knowledge that would connect that back to a species unless we provided it with an ontology, it being the language model here. In some cases, it doesn't quite get it right. It assigns taxa that maybe aren't quite appropriate. In some cases, that just comes down to the synonyms that are present in those ontologies. Like there's a synonym in the NCBI taxon that says, well, this is an appropriate ID for fish. Well, here that may not be exactly what we're going for. But things like chicken, it gets fine. It doesn't get all the relationships, but at least it gets eating. It links it to a Go term labeled feeding behavior. So definitely a lot more structure than what we started with. And if we had a variety of texts that looked a lot like this, well, we'd actually start getting a fairly decent collection of structured information. So this is the point at which I go into a little bit more detail about how we actually set up those structures and schemas. And they are all written in LinkML, which is essentially a data modeling language that our group has developed that with the goal of making setting up those data models and schemas a lot more flexible and essentially language agnostic. We write most of these schemas in the YAML meta language, but in practice you can start with JSON, you can start with OWL, you can start with essentially a tab delimited values of your actual data and set them up as a data schema. Because there are tools in the LinkML ecosystem that are set up for doing exactly this. But for OntoGPT and SPIRES, we'll usually start with them in YAML because that's very human readable and probably the easiest to write. Those schemas are, as I said, used to generate prompts. The prompts are passed to a language model, which essentially completes them. It defines a task for it to complete. What we get back is a completion payload that we then have to parse. We know that in many cases we're looking at part of that completion will define one extracted object. For example, if our text says deer in it or chicken, well, we're going to have a lot of other text in there besides just like deer and chicken. We need to be able to focus on that kind of individual instance. And try to ground it. And then if we can't ground it, then maybe we actually need to look a little bit more specific. And actually, we recursively generate prompts for subsets of our input text rather than just taking the entire text and trying to ground it as a whole. Although there are options to kind of skip this process as well. Another benefit here is that due to the structure of LinkML, we can translate our output to OWL. And if you're familiar with OWL, then one of the benefits of that structure is that we can reason over it. I'm not going to go into a whole lot of detail about that here, but given a sufficiently large collection of extracted items, you can essentially assemble your own ontology, or at least your set of classes in OWL. And you can reason over them to kind of fill in the blanks or learn about the properties that you'd expect to see. So if OWL is something you're familiar with, then this may be of interest to you. In practice, we can kind of define our more language agnostic schemas in a manner very much like this. This is one for chemical to disease relationships. Where we've said, okay, well, for each chemical to disease document, that's one document that may contain more than one set of relationships between chemicals and diseases. We know there may be some metadata associated with that document, like its publication details. But then for each one of those chemical disease relationships, we need to define details about, okay, well, what's a disease, what's a chemical, and what's the predicate that's going to link those particular chemicals and diseases. We can even add some additional qualifiers on top of that, whether we want to link them to another resource or not. And we can just be happy to extract the text in some cases. So what kind of data do we actually get in our extracted objects? So here's a relatively simple example where We've taken a paper describing beta-catenin's role in pathways, so in this case, signaling pathways, and its interactions with a protein from HSV1 or herpes simplex virus 1. So in this case, I've the template that I've used is one for Go causal activity models or Go CAMs, where we want to be able to extract things like, okay, well, we do want to know the names of genes that are present here, but we also want to know what activities that they're engaged in, what functions are they, and what kind of cellular processes, essentially the multiple levels of gene ontology. So essentially in one line, I can tell OntoGPT to parse this document with that template. By default, it uses GPT 3.5, which is the kind of the current default, but it can use GPT-4 as well. And what we see on the right here is what we get in our extracted object. We get sets of genes, organisms, and so on that contain the identifiers from the resources that we've specified. In this case, they're HGNC or human gene nomenclature IDs, because, well, this paper about human genes, but given a separate gene resource, it can link to those other IDs as well. Similarly, for things like activities and cellular processes, it's extracted the corresponding Go IDs. In some cases, it hasn't been able to find a match, so it's just given it the prefix auto and included the text that it found. For some of these, it may just be that, well, it's a slight mismatch to the category, or it just couldn't quite find the right match. So of course, this is certainly not limited to that particular use case. In this case, we have the figure legend from a study of the hypereic zone, which I believe is the zone around like stream beds and kind of the surrounding soils there and sediment. So this is from a 2020+1 study. So we can essentially take the text describing one of their figure legends. We use a template specifically designed for extracting environmental sample metadata. And that's exactly what we get in our extracted object. We can get the location that it's referring to. We can narrow down the specific environment and environmental material, and even additional variables about that. Like in this case, we can recognize, and I've included the labels for the named entities as well, which didn't have room for in the previous slide. We can realize things like, okay, well, we know we're working with sediment here. We're specifically talking about this hypereic zone. And we know that specifically we're working with water chemistry data. And so the label here is based on what we found in that text, but it'll connect back to at least what SPIRES has identified as an appropriate ID, and hopefully a consistent ID across multiple documents. So if you are interested in trying this out for yourself, you are certainly welcome to do so. It doesn't even require a whole lot of knowledge of Python, though it certainly helps. It is written in Python, requires 3.9 or greater. If you plan on cloning it directly out of the GitHub repo that is shown here, or you can just scan that QR code down there in the corner, then it requires the Poetry dependency toolkit, and there's instructions for installing that in the README. If you'd like to use the OpenAI models like GPT-3 and 4, it does require use of an OpenAI API key and paying for the individual calls. But we do have some options for running some open models that you can use for free, although they are notably quite a bit slower for now. That's a active area of study for how to get those to perform quite as well as these paid models. So this mentions here the ontology access kit as a requirement. You don't have to install that separately. If you install OntoGPT through Poetry or through pip, as if you're familiar with Python, and those are pretty common ways to install packages, it will install that as a dependency, so it'll be ready to go. So essentially, if you just want to test it out, you can try one of our basic templates here, and the extract command will do most of the work. That's kind of the bread and butter command for OntoGPT that runs the SPIRES method. And as I've been saying, essentially just pass it the name of a template. In this case, you pass it, the name of the template dot the name of a specific class in that template that you want to extract, and then you pass it the input as well. In this case, this is one of the the test fixture inputs that's included with the package. But of course, you can provide your own text, and you can certainly design your own templates, often just by basing them off the stock ones and just kind of modifying them as needed. Another handy thing is that OntoGPT will download all the necessary ontologies. It does that through Oak. So if you say, hey, I want to download the vertebrate breed ontology, it'll go and access that through the Obo Foundry, and it'll just download a local copy without any additional work necessary. So what's next for a project like this beyond actually just testing it out on some use cases? Well, we have a preprint out. Hopefully the published version of this will be out before the end of the year, but we're working on that. We'd like to try this out in some more use cases involving automated knowledge graph assembly. In many cases, that's really just focusing on a subset of the literature, extracting relations from it, and then integrating what we get with other knowledge sources that are out there. You can also predict additional relationships that you may have expected to find in literature, but didn't for whatever reason. So you gotta kind of get the best of both worlds. You get the domain knowledge from ontologies and knowledge bases. You get the more recent and experimental knowledge from the scientific literature, and you get to combine them in one place under a single data model. And that's pretty neat, especially when you can do it in a reproducible way. There are certain limitations that we're still working with. I alluded to the fact that, well, in many cases, we're still kind of reliant on OpenAI's somewhat mysterious models. We are moving towards open models for some cases, but yeah, as of right now, they don't perform quite as well as we'd expect, and they are relatively slow. There's also still kind of the ongoing problem of these hallucinations in some cases, and while the SPIRES method is designed to avoid them as much as possible, they do still crop up from time to time. So really, we'd like to not have those be present in data where we're trying to say things like this ID corresponds to this concept. We don't want to hallucinate that. We have a method with its own accompanying manuscript called Spin Doctor that's designed for doing gene enrichment analysis. More specifically, it produces a summary of a given set of genes. If you pass it, say, 10 to 15 genes, then it'll provide a text summary based on the individual text descriptions of each of them, which can be handy if you're trying to find something in common throughout those genes. This doesn't always work quite as well as we'd expect, so it's certainly not replacing human curators anytime soon, but there's some promising and probably helpful things that this method will be able to do in the meantime. And more broadly, we really just want to be able to do broad literature extraction and kind of, again, not replace biocurators in any way, but really help them along in the process, say by extracting from full text, whether those are from PubMed Central or otherwise, and given the schema saying, okay, well, here are the things of interest that we want to be able to extract, here are the kind of relationships that we want to extract. So given these texts, go at it. And we kind of get our initial set to work with. Speaking of larger sets, I should probably mention at this point that we have validated our results, at least in terms of relationship extraction, over the biocreative 5 chemical disease subset or the CDR task. So far, without further optimization, it seems like our method performs just slightly worse than the average F1 score on that task. The big plus being here that in those cases, they had to provide training data. We didn't. The language model is the training data. Okay, and I see there's a question about OntoGPT chatbot that I will get to in just a moment, because I'm almost at the end of my slides, and I want to be able to give that question as much attention as I think it deserves, which is a lot. And I think there's definitely some specific use cases for improving our integration with ag bio data resources and potentially including chatbots. Right now, OntoGPT uses this Oak module for its annotators and grounders. Oak is set up to work really well with ontologies from Obo Foundry and BioPortal, which means in some cases we can probably extend to using something like AgroPortal with relatively minimal effort. But we keep encountering use cases where we want to do things like extract plant or animal strains and genomes by name, but we just don't have quite the right resources for doing that to the level of accuracy that we want to be able to get. Even in cases where we do have the resources like the vertebrate breed ontology, we can extract livestock traits and breeds, but in real world text, we don't always get that level of detail. Sometimes the traits or the breeds or even the animal names themselves are used in kind of a very general fashion. Like you might just say like in bovines or in cattle, and then later refer using a more specific terminology. And we want to be able to capture those general terminology. And it's kind of challenging to do that consistently for right now. We don't have quite a solution for that. And I'm entirely willing to believe that there's a whole collection of other use cases that remain that I haven't even considered at this point. So at this point, I'll thank everybody in the group at Berkeley Lab, as well as additional folks who've worked on this SPIRES method and continue to work on OntoGPT. I welcome any questions by email, or feel free to check out my repositories on GitHub. And I'll certainly take any questions now. So I thank you all for your attention. And because we do have a question in the chat, I'll go ahead and address that first and give folks time to think up any questions that they may have as well. So yeah, it's in terms of chatbots, we actually have some other tools on the way that may actually be a little bit more chatbot-like. I'd say watch the space. We found that in some cases you can actually develop kind of plugins and modules for GPT that will have much of the same kind of functionality that we're looking for, while still building in the OpenAI infrastructure, which has its pros and cons. And we've tried that out with the Monarch Initiative data, and that seems like it's worked exceedingly well. It really does give you the nice benefit of working with a chatbot and that chat interface, while also having access to nice structured resources. So that there's certainly a space to have that operate as a chatbot as well, and say like, okay, given I have this kind of data, go get it from that source, and then give me the extracted text. So I'd like to think that we're certainly operating in areas where certainly the ChatGPT still isn't quite getting there, because if you just go to ChatGPT now and you ask it very similar questions like, hey, go to PubMed, get me the first 10,000 papers on this subject and summarize them based on the concepts that you find, it probably won't do that. At best, in some cases, it'll try to give you concepts, but it will hallucinate the IDs for them. So yeah, its results are a bit hit or miss, but we think that that's what we're kind of developing on top of. Do BERT models perform better than GPT? I think there are still some use cases. I saw someone recently, I don't recall exactly where, but did claim that they had a use case where BERT models perform better than GPT. And I think in some cases, when you're doing something like extraction of chemical names or gene names, I am willing to believe that there are cases where the task is defined specifically enough that BERT may actually do a little bit better. If only because those are the kind of tasks that lend themselves really well to those sequence based kind of tasks that those transformer models are really good at. Like, if you have a body of text where chemicals are usually drugs, or they're usually even something more specific, like maybe you're specifically talking about metabolites, then maybe they're always referred to in very similar contexts, and maybe that's the kind of detail that a BERT model can take advantage of and learn from. I'd like to think that those are cases where a GPT model could actually do quite well with a little help, whether it's something from OntoGPT, where we're using the outside knowledge sources for grounding, or just kind of tuning the model with additional examples, or even just giving it additional examples. Some of it really just comes down to this prompt engineering, where just very small changes to how you're phrasing or setting up that prompt can actually make a lot of difference in the results you get. So yeah, I think there's certainly some cases where kind of without further tweaking and updates, BERT models can perform better than GPT, but the GPT and the similar language models probably have a lot more flexibility for updating and making even further advances beyond what BERT's likely to do, whether it's named entity recognition or relationship extraction.
I also had a question. You mentioned that it would the ontology GTP that would download its own ontologies. Like, can you specify like plant ontologies or something else, or does it just download all ontologies that that's already directed to?
Oh yeah, if you mention it as an annotator in the schema, then the point at which it goes to annotate that kind of thing, like if you have a bunch of text with plant names, and you tell it, hey, use the plant ontology to annotate plant names, then the first time it finds something that the language model has identified as a plant name, it'll go try to ground it in that ontology. It'll download it as needed. So yeah, there are certainly cases where it could be easy to kind of download every possible ontology, but it's only going to do that if you specify them in the schema to begin with.
Hi, Harry. I have one more question, which is how much basic knowledge in terms of like installing software, or maybe, you need some bioinformatics knowledge, maybe you don't. Do you need to get started? Like you talk about defining a schema. What do I need to know? Like, assume I know very much or just about nothing. How much do I have to learn? What's our like activation energy to actually start implementing or trying this out?
Right. Yeah, and I think that's probably a good question for virtually any bioinformatics or data curation workflow. For this one in particular, yeah, I think it's always going to be more challenging than I would like. Right now, operating it doesn't require a whole lot of knowledge of Python. It does require knowledge of the command line and of LinkML, which has its own specific peculiarities. In practice, if you start with one of the existing kind of templates or the schemas that we have, and just kind of modify it in kind of like a piecemeal way, like say, okay, well, obviously this is a prompt description, or I see what ontology this is doing here, or this is using here, then yeah, it can be modified without really necessarily knowing a whole lot about YAML or LinkML. But I think one big assumption that we're making is that people are familiar with bio-ontologies to begin with. And certainly one thing that I've learned over the past few years is that there are areas of the bio-ontology space that just do not talk to each other. And sometimes it's, plant folks not talking with other folks. It's human disease folks not talking with other folks. So there's not, even if we know someone is familiar with a given collection of ontologies, they don't necessarily know what the appropriate ontologies would be for their given use case. Like someone came to me recently and they're like, I wanted to find a whole bunch of different data types. And they meant like clinical data types. But, I just, our whole group didn't have like one good set answer of like, okay, what ontology you could use, but you might want to use a whole collection of, some of this, some of this other one. And that's kind of the very general domain knowledge that I think will still remain a bit of a barrier given a particular set of needs. But yeah, it's the kind of thing where I'm hoping that over time we can, through some combination of documentation and live education and even just kind of setting up things like chatbots or Dockerized instances, or just more friendly interfaces, that we can gradually erode those barriers and reduce the activation energy.
And so just kind of bandwidth wise yourself or in your group, is there some capability for handholding. So let's say, oh, we'd like to try this out, but look at the documentation and maybe, you know, we've run into a wall. Are you maxed out? Is there some capability for like help?
Right. Yeah. And that's it. That's one of the nice things about this being kind of integrated with the rest of our other tool ecosystem is, yeah, we do have the ability to help with these use cases. Since, yeah, sometimes it's just a matter of like, hey, you know, if you understand LinkML a little bit better, then you can write these schemas for whatever you need. And we have workshops exactly for that. Or if it's a matter of like, you know, how do I just get started with this? We have people who can, you know, either point you to the right documentation or, yeah, provide it, you know, kind of one-on-one help as well. So that's something that we're happy to help with.
Thank you. That's it for me.
I think I see one more hand up.
Yeah, this is Subi. I would like to ask what you're trying to do with this at your group, and whether you've tried to do function annotations of genes with this, and if you can share any experience that you had with it would be helpful. Sorry if you mentioned that already and I missed it completely.
Oh, no. Yeah, it's I think there has been some interest in doing that for like I think primarily for microbial gene function. Like the model extraction, I think is some of the closest that we get right now. I think some of the areas where we've tried to leverage this a bit are in areas where we don't necessarily have a good set of model organisms, or cases where, you know, like maybe in we do have a good model organism for this particular gene or this pathway, but we don't in, you know, our target organism. We haven't had a lot of direct use cases on that yet, but it's definitely been discussed. And yeah, I think in some ways, yeah, it really just comes down to modifying some of our existing schemas to correspond to the kind of target text that that we're working with. But yeah, it's the short answer is, yeah, it's been discussed, but yeah, no direct implementations as of just yet. And I see a question about workshops. Yeah, we don't currently have any OntoGPT workshops scheduled, but that might be an interesting thing to start doing. If I wanted to spread that idea to this group in particular, is there a good mechanism for doing that?
Definitely yes, yeah. We have a Slack channel, and then a number of other ways like emails, and we're thinking about doing doing newsletters eventually soon too. I also just wanted to mention, and I probably could just do an email too, like I think a number of us would be happy, I know at least myself would be happy to be like almost like a beta tester for you, since I know some bioinformatics, I know the data curation part, but I don't know a lot about the language model, the large language models. I'm really good with the terminal, you know, command line and all that various stuff, but like jumping from one to the other, I don't have that knowledge. So it could be one of those things where, like, especially for documentation, you know, like how I try to go through some of the documentation, I'm like, I don't get how you get from step A to step B.
Yeah.
It might be something like that. I think a lot of, especially myself, I'd be happy to help with this because I can see this being a really good resource for bound creation in the total. So
yeah, that's I'm glad to hear that. And yeah, all feedback is both appreciated and necessary.
Mm-hmm. Any more questions? We have 3 more minutes left. All right, if no more questions, let's give a round of applause again for Harry. Thank you so much for coming. This was a great talk. And for the people who missed it, we will be putting it on our YouTube video later, probably later this week. So again, thank you.